account. Umeweka mangu for one year. Style. Sasa anaanza kuwadhibiti watoto. Ni nani? Kibia pale. No, enda pale.
Mwalimu anaitwa nani?
Okay, yeah, I'm now the host. Okay, good morning, everyone. I hope you can all see me and hear me. We are just waiting for a few of us, more of the students to join before we can start. So we are starting at exactly 11. At exactly 11, we'll be able to start. My charger is inside the camera. Okay, good morning once again, everyone. We are just about to start. Now, even as more of you are still coming in, I want to take this opportunity to welcome you to our class today. We are meeting for the first time. Last week you had course that is biology syllabus, and the topic is the cell. The topic is the cell. Okay, thank you. I've just been reminded that you had looked at okay, so I've just been reminded that the topic was introduced in the last class. So it means that we will just proceed from where you stopped. You are looking at the the parts of the light microscope. The parts of the light microscope. That is where you reached last time with Madam. So I think it is important for us to remind ourselves of what you learned in the last class, just as a recap before we proceed. Now, the topic is the cell, and I believe you all learned. You all learned. So, okay. Now, we'll start by looking at the definition of the cell, which you did last time. And I believe you can all see my screen. I'm already sharing my screen. Now, we all know that all living organisms are made up of cells. That means, therefore, that the cell is the basic unit of any living organisms. So when I say the basic unit of any living organism. That simply means that this is the least unit that makes up the structures of an organism. So the structures within the body of organisms, both plants and animals, are made up of the cell. So the cell is the basic unit of an organism. Now, it is very evident that some organisms are so small and others are very big. Now, some organisms are only made up of a single cell. Now, those organisms that are made up of a single cell are called unicellular organisms, while those ones that are made up of multiple or many cells are called multicellular organisms. Now, I remember in the exam that you did recently, there was a question that was asking you to define unicellular and multicellular organisms. Now, I believe you all can see and you've all learned that unicellular organisms are those organisms that are made up of a single cell, or they're all sometimes called single-celled organisms. And examples of such organisms include amoeba. And those ones that are made up of many cells are called multicellular organisms, from the word multi. Multi, as you know, multi means many. So multicellular organisms are those organisms that are made up of very many cells. Examples include these big plants like the beans, maize. We also have animals like cows, donkeys, and even human beings are multicellular organisms. Okay, so if you didn't get the question right in the exam, I believe you can now see what you should have written in the exam, what should have included in your answer in the exam. So the cells, as we know, cells are very, very small structures. They are very small that we cannot be able to see them using unaided eyes or using our naked eyes. They are very tiny. They're so small that we can't see them using our naked eyes. And therefore, for us to see and observe the structures of a cell, we can use an instrument which is called the microscope. So the microscope is an instrument that is used to magnify. So when we use a microscope to observe the cell, we can be able to see it clearly and identify some of its structures. So microscope basically helps us to identify. It is not only used to magnify, but even to bring it can be used to distinguish two close objects that cannot be seen clearly. So even as you look at the structure of the cell, you are able to see some of the structures that are not clear and can be distinguished with the help of a microscope. Now, when talking about microscope, we have got two main types of microscope. One, we have what is called the light microscope, and the other one, we have what is called the electron microscope. Now, we will start by looking at the light microscope, which you looked at in your last class. And like I said, I'm just going to be giving a highlight of what it is. Now, the light microscope, like I said, the microscope is an instrument that is used to magnify objects. It is also used to manipulate the resolution of the image. That means it is used to show up the finer details. It is used to show up the finer details. Those details of an image or an object that cannot be seen clearly, they can be resolved with the help of a microscope. Now, the reason why it is called a light microscope is because it uses light for illumination of the specimen that is being viewed. Okay, it uses light. That is why we call it the light microscope. Now, we will also light and electron microscope later on. I think after we shall have looked at the light microscope, we'll be able to compare and the electron microscope. Great. Now, There is a diagram here that shows a light microscope, but I want to show you a better diagram of the light microscope. So just a minute, I'll be able to get you a better diagram of the light microscope here. Now you can all see my screen here. What you can see on the screen is a structure of a light microscope. The structure of a light microscope here. And I remember before we went home for the break, we had very many practicals in the lab where we were able to see the light microscope and even use it to observe some specimens. Now, if you this is exactly what we all saw in the lab. This is exactly what we all saw in the lab, and we were able to use the light microscope to observe some specimens that were provided during that time. Now, if you look at this diagram here, this is the light microscope. It has got the following parts. We have got what is called the eyepiece here. The eyepiece here, the eyepiece lens, which is found here. We have got this tube, and the tube is called an ocular tube. Ocular tube. We have got the coarse adjustment knob here, the fine adjustment knob, which is just next to the coarse adjustment knob here. Then we have, okay, the tube here, this is the body tube. This is the body tube, sorry. The body tube, and I know we will be able to look at the functions of these various parts. And then we have got what is called the lens are attached. All the objective lenses here are joined here. We have got the arm, we have got the condenser, we have got the diaphragm. Now the diaphragm is just found here, and then the condenser, as you know, is found somewhere just above the diaphragm. Then we have got the stage where we always mount our slides. When we're preparing, after preparing a slide, it is always mounted on the stage just so that the specimen can be easily observed. Then we have got the light source, which is usually the mirror. Then we have got the base here. Now I know some of the this is just a diagram which I was able to obtain from the from some website. It is including some parts that I know we never mentioned earlier, but you don't have to mind as long as you know the main parts of a microscope, then we are good to go. So this is a diagram that shows a light microscope. It basically has a basic structure that looks like the one that I'm displaying on my screen. So I want us to very first move and identify some of the functions of the various parts of the light microscope. Thereafter, we'll be able to look at how to handle the microscope, and then without wasting time, we'll also be able to move forward and look at the structures of the plant and animal cells as seen under the light microscope. And like I said, these are some of the things that you handled last week, so we don't have to waste much time on them. So we are back here. The functions of the parts of a microscope. Now we have the first part, which is called the eyepiece, or what we call the ocular lens. The eyepiece of the eyepiece, or what we call the ocular lens. Now the eyepiece of the ocular lens, it contains a lens which contributes to the magnification of the specimen under view. It contains a lens which contributes to the magnification of the specimen under view. Now, when you talk about the word magnification, now later on you'll be able to see how to obtain or how to calculate magnification. And what you need to know is that the total magnification of the specimen that is under view is contributed to by all the lenses that we have here. Now, we'll be able to see that we have got two lenses here. We have got the eyepiece lens and the objective lens, and both of them contribute to magnification. So we'll be able to see how to get the total magnification of the specimen that is under view. So the eyepiece of the ocular lens, we've said it contains a lens which contributes to the magnification of the specimen under view. The second part, which is just below the eyepiece, is called the body tube. The body tube. Now, the body tube, it supports the eyepiece and the revolving nose piece. It supports the eyepiece and the revolving nose piece. As you can see, let me just show you where we have the body tube. The body tube here. This is the body tube. This is the body tube here. I believe you can all see my cursor. Now, the body tube, as you can see, it joins the eyepiece lens and the revolving nose piece here. Now, the revolving nose piece, this is where we have got the objective lens attached, as you know, the revolving nose piece here. Then, so this point that joins the eyepiece lens and the revolving nose piece, that is where we have the body tube. So the function of the body tube is to support the eyepiece and the revolving nose piece, to support the eyepiece lens and the revolving nose piece. The third part is the objective lens. And as you can see, we are following them in that order from the top to the bottom. Now, the objective lens are usually, okay, sometimes you'll find a microscope that has got more than one objective lens. Now, we have the low power objective lens and the high power objective lens. Sometimes you can even find a microscope that has got three objective lenses. So both of them are used under different circumstances, especially when you're trying to find when you want to obtain a clear magnification or a better view of the specimen that is being observed. You'll always turn the high power objective lens or the low power objective lens so that you can be able to come up with a clear view of the specimen. So when you're asked, now when talking about the function of the objective lens, now the objective lens are usually fixed on the rotating, or what we call the revolving nose piece, and it brings the image into focus and magnifies it. It brings the image into focus and then it magnifies it. That is the objective lens. The coarse adjustment knob. Now, and I want you to get the clear differences between the coarse adjustment knob and the fine adjustment knob. Let the difference come out clearly, because in the last exam that you did, we were able we saw some students trying to confuse the function of the coarse and the fine adjustment knobs. They are almost close to one another, but even in size, you'll be able to see that the coarse adjustment knob is bigger in size as compared to the fine adjustment knob, and they perform different functions. Though the functions are almost the same, but they are totally very different. So the coarse adjustment knob, it raises or lowers the body tube for longer distance to bring the image into focus, and not just focus, but into rough focus. Now, on my screen here was not, let me just add the word rough so that you can be able to know what I mean. It raises or lowers the body tube for longer distance to bring the image into rough focus. Now, I want to highlight something here that is very important. The function is to raise or lower. Some of you say to raise and lower. No, I think even from the English itself, the two words are contrary. You cannot raise and lower at the same time. It can either raise or lower. Let it be known to you today that it is raising or lowering, not raising and lowering. That is wrong. It is raise or lower the body tube for longer distances to bring the image into rough focus. The fine adjustment knob, it raises or lowers the body tube through smaller distances. Now you can see the difference. The first one there was used to raise or lower the body tube for longer distances, but this one here is to raise or lower the body tube through smaller distances to bring the image into fine focus. I'm just adding the word fine there so that for those of you are taking note, you can write the right thing, into fine focus. And then we have got the arm or the limbs. Now, if the arm or the limb is that part that is usually held together with the base so that you can, when carrying the microscope, that part that is usually held. And we said that when carrying a microscope, you must use your 2 hands. So the arm or the limb is that part that supports the body tube and the stage. It connects the body tube and the stage. And it is also used when carrying the light microscope. Then we have got just below the arm and the limb, we have got the stage. We have got the stage there. And the stage is a platform where the specimen on the slide is placed. It is a platform where the specimen on the slide is placed. Great. Now, then we have got the clips. The clips are usually mounted on the stage. If you look at the stage, now assume this is the stage. I don't know whether you can all see my hand, but then if this is the stage, on the stage we have got those 2 metallic, they look like pins, but they're always 2. One on the they are both on the ends of the stage. They're usually mounted there. Now these clips, their function, they are used to hold the slides. Once you've prepared your slide, and we all know that slide is usually prepared separately before it is mounted on the microscope, before it is mounted on the stage of the microscope. So when you have your slide prepared separately, you can bring it now. When you want to view the specimen, now you bring it to this and put it to the stage, and then you mount it on the stage, then hold it in place using the clips. So the function of the clips is to hold the slide in position, to hold the slide in position. Great. Number 9, we have this part that is called the condenser. The function of the condenser is to concentrate light on the object on the stage, to concentrate light on the object on the stage. Now, the condenser is always found above the diaphragm, which is the next part of the microscope we are looking at. Now, it concentrates the light. The light comes from the mirror, but it is channeled through the diaphragm. So the function of the condenser is to concentrate that light so that the specimen that is being viewed can be easily seen. So the condenser, it concentrates the light on the object on the stage. Remember, we are talking about the light microscope, and light is very key here. That is why even when we'll be looking at how to handle the light microscope, it should always be faced towards the direction where the light is coming from, because light is what is used to illuminate the object that is being viewed. Then we have the diaphragm. The diaphragm is that part of the microscope that regulates the amount of light passing through the condenser to illuminate the specimen. It regulates the amount of light that is passing through the condenser to illuminate the specimen. Then we have the mirror. Now the mirror reflects the light. Its function is to reflect the light through the condenser to the object. The light is very important here. So the mirror here reflects the light from the source towards the condenser, and this light is regulated by the diaphragm. So I believe you're able to see how these parts of the microscope, how these parts of the microscope are positioned. They're positioned in such a way that the diaphragm is below the condenser because it regulates the light which gets into the condenser, and the mirror is below the diaphragm. So the mirror is usually very flexible, so that it can be made to face the direction of the source of light. It is usually very, very flexible. And I believe you also that when we had that practical before you went home. Now, the last part that is not included here is the base. The base, as you know, it provides support and stability to the microscope. It provides support and stability to the microscope. And usually, the base is always made up of a very heavy material. The material that is used to make the base is usually very, very a heavy one, so that it provides support and stability to the microscope, so that while using the microscope, it does not fall. It remains stable until you're done using the microscope. So those are some of the parts of the light microscope. And I know I've looked at them very fast because there are some of the things that you learned in your last class. I was just doing a recap so that no one is left behind. Now, handling care of a microscope. There are some simple rules that are usually followed when handling and taking care of a microscope. And they include Number one, it is important for you to always use both hands when carrying a microscope. And how do you use these both hands? One should be used to hold the base, and the other one is used to hold the limb or the arm. And like I said that the arm or the limb is that part that is usually held together with the base when carrying a microscope. So when each and every time when you're handling a microscope, you should use both hands, one holding the base and the other one holding the limb. Number two, when using the microscope, never place the microscope too close to the edge of the working bench or table. Not place the microscope too close to the edge of the working bench or table. Why do we say that? Because it can easily fall. When you put it towards the edge or too close to the edge of the working bench or table, this microscope can slip and fall. And remember, this is a very, very expensive instrument to obtain. So when it falls, it can easily get damaged, and so it will be meaningless. So it is important to always avoid putting the microscope very close to the edge of the working bench or table. Number three, do not touch the mirror and the lenses with your finger. Do not touch the mirror and the lenses with your finger. Why do we say this? Because these are very, very delicate parts of the microscope, the mirror and the lenses. Now the lenses are very, very key while using the microscope because they're the ones that are used to get the magnification of the specimen. So when you touch the lenses with your finger, they can easily get dirty, and some of them can be easily broken. So they can be easily broken when handled the wrong way. So avoid touching the lenses and the mirror using your fingers. Now, what should you do in cases where the lenses and the mirror are dirty and they need to be cleaned? So dirty lenses should be cleaned using a special soft lens tissue paper, or that tissue paper, the normal tissue paper, which is moistened or softened using ethanol. So in case of dirty lenses, you should clean them using a special soft lens tissue paper or a tissue paper that has been moistened using ethanol. Okay. Then another way of handling the microscope. Other parts of the microscope, when maybe they're dirty, they can be cleaned using a soft cloth or tissue paper. That is for the other parts. Leave alone the lenses and the mirror. Those other parts can be cleaned using a soft cloth or a tissue paper. Okay. Then finally, do not wet any part of the microscope. Remember, the microscope is made up, some parts are made up of steel, iron. These are materials that can easily rust. And you know, I think you've learned what causes rust, what rust is from your chemistry classes. Now, rust is always there whenever a metallic surface, either iron or iron, is reacts to water. So we should avoid touching any parts of the microscope using our wet hands, or maybe spilling water on the microscope, because it can easily get rust. Okay. Now, another way of handling the microscope: make sure the low power objective lens click into position in line with the eyepiece before and after use. Make sure the low power objective lens click into position in line with the eyepiece lens before and after use. And I will ask a very simple question at the end of the class: why do we have to always leave our why do we always ensure that before and after use we have to ensure that the low power objective lens click in position in line with the eyepiece lens? Why do you think that has to be done? I hope you'll get me the answer at the end of the class. This is something that I think we did last time. Then after use, always clean and store the microscope in a safe place, which is free from moisture and dust. Just be responsible, clean and store the microscope in a safe place, which is free from moisture and dust. Number 9, when focusing, always move the lenses upwards to avoid breaking the microscope slide and the cover slip. Now, that is for the objective lens. Try and move them upwards. Try and move them upwards, not downwards towards the stage where we have got the slide. Remember, the slides are made of glasses, so these glasses can easily break, especially when you move the lenses towards the slide. So every time you're focusing, move the lenses upwards to avoid breaking the microscope slides and even the cover slip that is used to cover the specimen on stage. Okay. Now, the next thing that we're looking at is using the light microscope. And like I said, these are things that you had covered in the last class, and so we don't have to really waste time on them. We're just trying to ensure that no one is left behind. Now, when using the light microscope, how do you use the light microscope now? When you have a specimen that you'd want to observe and you'd want to use a light microscope, there are very simple basic steps that are important for you to follow. Remember, this is the light microscope, and the reason why we call it the light microscope is because it uses light to illuminate. It uses light to illuminate the specimen that is being observed. Okay, so using the light microscope, the following are some of the steps or procedure to follow when using the light microscope. Number one, place the microscope on the bench with the stage facing the source of light. Place the microscope on the bench with the stage facing the source of light. Now, remember, the stage should always face the source of light. And just below the stage, we have got the mirror. And the mirror is the one that reflects the light from the source through the diaphragm to the condenser, so that the condenser now will now concentrate it on the specimen that is being observed on the stage. So the stage must always face the source of the light. Number two, set the low power objective lens in focusing position. Set the low power objective lens in focusing position. And I think I've just mentioned this. It is always important to always ensure that the eyepiece, the lenses are in line, are in the same line as the position of the low power objective lens. Now, it is always important for you to set that. Then look down through the eyepiece and adjust the mirror so that enough light is reflected from the source into the microscope. Look down through the eyepiece, and that is why we said it. You can only look down through the eyepiece using your one eye, not all of them at the same time. Just one eye. You can use your one eye to look down through the eyepiece and adjust the mirror. So the reason why you're adjusting the mirror is so that you can have maximum light reflected. You can have maximum light reflected into the specimen that is being observed there. So look down through the eyepiece and adjust the mirror so that enough light is reflected from the source into the microscope. Okay. Now, while viewing from the side of the microscope, turn the coarse adjustment knob until the objective lens and the slides are very close. Now, you said you remember the function of the coarse adjustment knob. Said that it is used to raise or lower the. It is used to raise or lower the what? Sorry. Okay. For us to have that raw focus first, we are going to be using the coarse adjustment knob to raise and lower the body tubes, and then as we find the raw focus, and this is usually done by you turn the coarse adjustment knob until the objective lens which are held on the revolving nosepiece there are close to the slide. And we said that you should not take them very close to the slide because they can interfere with the slides. The slides are made of glass, and therefore they can be easily interfered with by the objective lens. So you just take them close to the slide, but do not allow the objective lens to touch the slides, because the slides can be easily broken. And then place the prepared specimen on the microscope slide, then place it at the center of the stage. Now, as you carry on with learning biology, I think it is in the same topic where we learned how to prepare slides. Slides are usually prepared. Now, when we talk about slide is made this is where we put the specimen, and then we cover it using the cover slips. Cover slips are some tiny pieces of small glasses that are used to cover the specimen. So place the prepared specimen on the microscope slide, then put it at the center of the stage. The slides are placed at the center of the stage, and then they are held in position using we have got those clips, the clips that are usually found on the stage. So the clips are used to hold in position the slides that are to be observed using the microscope. And then looking through the eyepiece, turn the coarse adjustment knob, moving the objective lens away from the microscope slide until the specimen comes in view. You use the coarse adjustment knob. I mean, this is now just to bring that rough focus, the one that we mentioned earlier when we were looking at the function of the coarse adjustment knob. So you use the coarse adjustment knob to move the objective lens. As we know that I used to raise or lower the objective lens through longer distances to bring the image into rough focus. So you just use the coarse adjustment knob as you move the objective lens away from the microscope slide until the specimen comes in view. And this is the rough view, Now bring the specimen in sharp focus using the fine adjustment knob. Bring the specimen into sharp focus using the fine adjustment knob. Now that is where now we use the fine adjustment knob now to bring the image into sharp focus. Now we are supposed to adjust the amount of light passing through the diaphragm to obtain a better view of the specimen, because light is our main source of is what we use here to illuminate. So we adjust the amount of light passing through the diaphragm to obtain a better view of the specimen. And then we are supposed to adjust the high power objective lens. Like I said, objective lens, there are very many types of objective lens. You have got the high power objective lens and the low power objective lens. So high power objective lens, they're the ones that they have got a very higher magnification as compared to the low power objective lens, depending on the type of specimen that you are viewing there. So you adjust to high power objective lens, and then you're supposed to always keep both eyes open when using the microscope with one eyepiece. Just supposed to keep both eyes open, because I think there's a coordination between the eyes. Some people may find it difficult to close one eye and open the other one. So just keep all eyes open when using the microscope with one eyepiece. Now, those microscopes that have got two eyepiece lens, but those ones that have got just one eyepiece. So even when using the one that has got one eyepiece, just keep both eyes open. But you cannot use both eyes while observing while observing a specimen using one eyepiece. The eyepiece lens is very, very small, that we can't use two eyes to view at the same time. Okay. Now, the next thing that I wanted to highlight from your last class is magnification. Now, after coming up with the specimen on the stage, now you're viewed. Now, sometimes you can be asked to calculate the magnification of the object. How to calculate the magnification of the object? Now, when you're asked magnification, I think we mentioned earlier, even when you're looking at the topic classification, we were able to calculate magnification. That is when you're using the hand lens. The hand lens are also used to magnify tiny objects. But again, when talking about magnification of the microscope, now magnification of the microscope is totally different from that one of the light microscope, because here we are using the lenses that are used to magnify the objects. And magnification refers to the number of times an object is enlarged. So to find the number of times an object is enlarged using a microscope, we are going to use the magnification of the lenses that are used to magnify the object. And I said earlier that we have got two main lenses here. One, we have the eyepiece lens. And then we also have the objective lens. So the magnification of the objective lens and the magnification of the eyepiece lens are brought together to give us the total magnification of the microscope. I mean, not of the microscope, but of the object that is viewed using the microscope. So if you're asked to calculate the magnification of the object, simply use the eyepiece lens magnification and the objective lens magnification. And here we will only use depending on the objective lens that you use. We can sometimes use high power objective lens or low power objective lens. So when, let's say, for example, an example is given there on my screen. If the eyepiece lens magnification is times 10 and the objective lens, the low power objective lens is also times 10, then you are told to calculate the total magnification of the object. What you need to do is to find magnification, just multiply the magnification of the eyepiece lens and the magnification of the objective lens. So that is times 10 times 10. And then do not forget the sign, the multiplication sign there. This X before the 100 is very important. Now, I was going through the exam that you did, and maybe if you allow me to just show you the exam that you did, there's a question about magnification that a good number of students did not find right. So this is the right time to, I think, address that here. This is the exam that you did. The question about magnification was number 5. So there's the first part of that question was asking you to distinguish between magnification and resolution. Magnification and resolution. Now we are going to be able to see that just after you're done with magnification, when we introduce the word resolution. Now, I was interested in part B of number 5, where you are given the eyepiece lens magnification, the objective lens magnification, and the total magnification. Now, some of you failed this question because of very simple mistake here. And this is what some of you did. First of all, you need to know that every time you're talking about magnification, do not forget this symbol, X, this multiplication symbol here, every time. So for this case, we have said that for us to get the total magnification, we multiply the eyepiece lens magnification by the objective lens magnification. And from your basic mathematics knowledge, if you're given the eyepiece lens magnification and the total magnification, then you are asked to find the objective lens magnification. What you need to do is just divide the total magnification by the by the eyepiece lens magnification to get the objective lens magnification. So this one, you are supposed to divide 600 by 30. And if you divide 600 by 30, you get 20. So the answer here should have been times 20. The second one here was times 14, which is the magnification of the eyepiece lens, and the magnification of the objective lens was times 5. So total magnification would be calculated by multiplying the eyepiece lens magnification by the objective lens magnification. And if you do that, you get times 14 times 5 gives you times 70. And then the last one here, which we were given the objective lens and the total magnification here. Then you are told to find the eyepiece lens magnification. So that should have been, you divide times 2000 by times 40. If you divide 2000 by 40, you get 50. So the eyepiece lens magnification here should have been times 50. I believe we are all at that point. There are very many questions from this topic that some of you did not do well, and I would want to try and emphasize on those areas. So if you still find difficulties there, you can ask questions. And I would prefer that if there is any question that is very, very pressing, you can use my chat there. Okay. So let me just get you back to where we are. Great. So that is all about magnification, or how to find magnification using the light microscope. If you are told to find the magnification of the object that is being viewed using the microscope, then use the magnification of the eyepiece lens and that of the objective lens, and then you come up with that. Okay. Now, we said earlier that we have got two types of microscope. We have the light microscope and the electron microscope. Just want to give a highlight of the electron microscope because we know it is not commonly used. And this is what you need to know about the electron microscope. The electron microscope, it uses a beam of electrons to illuminate the object, and that is why we call it the electron microscope. It uses a beam of light. It uses a beam of light to illuminate the object. Sorry, beam of electrons, not beam of light. Beam of electrons to illuminate the object. And it is usually very powerful than the light microscope. Now, because it uses the beam of electrons, it becomes difficult for us to view live specimen with it. We cannot view live specimen using the electron microscope. In most cases, it is used to view dead specimens. And it has got more sophisticated parts, and it has got a high resolving power. Now, when we talk about the word resolving power, this refers to the ability to distinguish between two close points. The ability to distinguish between two close points. That is the resolving power. So if you hear the word resolving power, just know that it refers to the ability to distinguish between two close parts. And I want to believe that you're making notes. I want to believe that you're taking notes wherever you are so that you are not left behind and you can use this for your revision. Now, very first, let us try and look at some of the differences between the light and the electron microscope. Differences between the light and the electron microscope. Number one, the light microscope uses light to illuminate the object. Light microscope uses beam of light to illuminate the object, while the electron microscope uses the beam of electrons to illuminate the object. The electron microscope uses the beam of electrons to illuminate the object. Now, I've seen a good number of students trying to raise their hands. I would prefer that if there is any question or any comment, you can just notify me using the private chat. Just notify me using the private chat. That is, I've seen Kelly K has raised his hand. Just notify me using the, okay. Number two, the light microscope. The light microscope has a low resolving power. The light microscope has got a low resolving power, while the electron microscope has a high resolving power. While the electron microscope has high resolving power. Number three, the light microscope has a low magnification. The light microscope has got a low magnification, while the electron microscope has got a higher magnification. Number four, finally, the light microscope is used to view both live and dead specimens. The light microscope can be used to view both live and dead specimens, while the electron microscope is only used to view the dead specimens. The electron microscope is only used to view the dead specimen. Okay. The light microscope uses glass lenses. The light microscope uses glass lenses to magnify. The electron microscope uses electromagnets to magnify. It uses electromagnets to magnify. Then in light microscope, the specimen is usually viewed through the eyepiece, while in the electron microscope, the specimen is viewed on a screen, or what we call a photographic plate. A photographic plate. Great. So that is, I think up to that point, those are some of the things that you looked at in your last class. And up to this point now, I want us to move very straight for and look at the various cell structures are seen under the light microscope. Cell structures are seen under the light microscope. Now we are going to be looking at the cell structure of the animal and the plant cells. The structures of the animal and plant cells are seen under the light microscope. The structures of the cell are seen under the light microscope for both plant and animal cells. Now, it is important for you to note that those structures which are found within the cell, those structures that are found within the cell are known as organelles. Organelles are structures which are found within the cell. Now, some of the organelles are seen under the light microscope. Now, when using the light microscope, we may not be able to see very distinct cell organelles as compared to the electron microscope. So the light microscope will just give us very few organelles as compared to the electron microscope. Now, some of the organelles are seen under the light microscope include, number one, we have what is called the cellulose cell wall. And the cellulose cell wall is only found in the plant cells and not in the animal cell. The cellulose cell wall, we have the cell membrane, which is found in both the plant and animal cells. We have got the cytoplasm, we have the nucleus, we have the vacuole, and we have the chloroplast. So those are some of the cell organelles that are usually shown that can be seen when using the light microscope. Now, unfortunately, I don't have any diagram of the plant and an animal cell as seen under the light microscope. But I want to promise that in our next class, I will find two diagrams, one for the plant cell and the other one for the animal cell, as seen under the light microscope. As seen under the light microscope. Okay. Now, organelles perform specific functions within the cell. The cell organelles perform specific functions within the cell. And we are going to look at some of these functions which are performed by these cell organelles. Now, I want to say again that because I don't have any diagram here for the structures of a cell as seen under the light microscope, I'm unable to show you any, but I'm going to avail some in our next class so that you can be able to see the structure of a plant cell as seen under the light microscope and the structure of a cell as seen under the an animal cell as seen under the light microscope. Those ones I will try and avail them. And even the ones of the electron microscope, both the plant and animal cell, I'll be able to find you them. Okay. The first structure that you're going to see is the cell wall. The cell wall. And the cell wall, as we said, it is found only in plant cells where it forms the outer boundary of the cell. It is only found in the plant cell where it forms the outer boundary of the cell. It is composed of cellulose fiber. It is composed of cellulose fiber. And let me just try annotating the word cellulose here. Cellulose fiber. Now, in most cases, you've always seen the cell wall referred to as cellulose cell wall. Now, we call it the cellulose cell wall because it is made up of cellulose fiber. Now, this cellulose fiber, the cellulose fibers here, they provide an additional strength to the cell wall. They provide an additional strength to the cell wall. And it's a very interesting thing that we'll see here. I think when we're looking at the next topic, which is cell physiology, we'll see the roles that are played by these cellulose cell walls in plant cells. Now, these cellulose fibers, like I said, they provide additional strength to the wall. They strengthen the cell wall. They make the cell wall, they make the cell walls to be very, very strong. Another thing that you need to know about the cell wall, the cell walls are usually fully permeable. They're usually fully permeable. Now, when I say fully permeable, it means that they allow water and mineral salts to get into the cell. They are fully permeable. And note this word fully permeable, because we are going to be able to identify the difference between the cell wall and the cell membrane later on. So the word permeable here is very, very key. So the cell wall is fully permeable to water and mineral salts. Note that. Okay? Note that kindly. Now, the functions of the cell wall. Having looked at the structure of the cell wall, you've said that the cell wall is usually found, it is the one that is found out as the outer boundary. It is found on the outer of the plant cell, and it forms the outer boundary of the cell. And we've said that it is made up of cellulose fibers, which gives it, which strengthens it. And it is also fully permeable. It is fully permeable to water and other materials such as mineral salts and many other materials that get into the cell and leave the cell. So the functions of the cell wall. Number one, the first function of the cell wall, it gives the plant cell a definite or a fixed shape. It gives the plant cell a definite or a fixed shape. If you look at the plant cell has got a definite shape as compared to the animal cell. The animal cell can take any shape. It can take any shape, but the plant cell has got a definite shape. So the definite shape in the plant cell is contributed to by the cell walls which are found in the plant cells. Number two, the second function of a cell wall is to provide mechanical support to the cell, to provide mechanical support to the what? To the cell. mechanical support here, it provides mechanical support because it is strengthened using the cellulose. So the cellulose fibers, they make the cell wall to be very strong so that it can provide that mechanical support. Number 3, it provides protection against mechanical injury. It provides protection against mechanical injury because it's the one that is found on the outward, on the outer part of the cell. It provides protection against mechanical injury. Number 4, it allows gases, water, and other substances to pass through it due to permeability to water and mineral salts. Now, we said that the cell wall is fully permeable. Fully permeable means that it can allow anything to pass through it because it is because of the nature of it, because of the nature of the cell wall. I mean, it allows any material to pass through it. So it allows gases, water, and other substances to pass through it due to permeability to water and mineral salts. So gases, water, and other substances all pass through the cell wall, either from outside of the cell to inside of the cell, or from inside of the cell to outside of the cell. So the cell wall will always allow these materials to pass through. Okay. Now, as we proceed, we're looking at the cell membrane, or what you call the plasma membrane. The cell membrane or the plasma membrane. Now, the cell membrane or the plasma membrane, this is a cell structure. Maybe to let you know what a membrane is, a membrane is a surface structure which is found on the surface of a cell or of a cell organelle. We have got cell organelles which have got membranes on them. And even the cell itself has got a cell membrane. So the cell membrane is that surface structure which is found on the surface of a cell. It can also be referred to as the plasma membrane. So the cell membrane is found in both the animal and the plant cells, where it encloses the entire cell. All the cell organelles are enclosed using the cell membrane. And it is interesting to note that the structure of a cell membrane consists of 3 layers. Now, these 3 layers include, 1, we have got 2 protein layers. We have got 2 phospholipid layers, not 2 protein layers. We have got 2 phospholipid layers. Now, when we talk about the word phospholipid here, let me just highlight this so that I can explain more of it. We have what is called a sorry for that. Now we're talking about a phospholipid layer. And the word phospholipid is made up of 2 words. 1, phosphate, and lipids. So a phospholipid layer is just a layer that has been made up of phosphates and lipids. So we have got 2 phospholipid layers. One on the inner part of the cell membrane and the other one on the outer part of the cell membrane. So in between the phospholipid layers, we have got a protein layer. Okay. Let me just find out something here before we proceed. I think it is important to show you the diagram of a cell membrane. Just trying to show you the structure of a cell membrane here in less than a minute's time. just finding out the structure of a cell membrane so that for those of you who have never seen what a cell membrane looks like and be able to see it right now. Okay. So here is my great. Now look at this. This is a very, very complex structure of a cell membrane. Now the cell membrane here, as you can see, we have got these two layers of protein. Now, sorry, I think I made a mistake there. I said two phospholipid layers. Now we have, it is not two phospholipid layers, it is a single phospholipid layer, which is found between two protein layers. Note that correction kindly. Two protein layers sandwiching one phospholipid layer. Now, these balls that look, these balls here that look, the orange balls here, the orange balls here, these are protein layer. And even down here, as you can see, we have got another protein layer. So we have got these two protein layers down here, one here and the other one on the outer part. So in between here, we have got the phospholipid layer. The phospholipid layer is found in between the two protein layers here. And then another thing that you need to note, as you can see, this is a very complex diagram. I know some of you have never seen this before. Some of you have never seen this before. And from what you said earlier, if you look at the structure of a cell membrane, then in between we have got some tiny openings. In between you have got some tiny openings. We have got some tiny openings which we call the pores. It is made up of the pores, and the function of these pores is to regulate the movement of substances into and out of the cell. Now the pores are very tiny, such that they only allow those substances of the right size, those materials or particles of the right size, those ones that are small that and can pass through the pores are the ones that are allowed to get in. So these materials, because of their sizes and because of the size of the pore, we say that because of their sizes and the size of the pore, that is why we say that the cell membrane here has got a very unique property as compared to that of the cell wall. And we say that it is what? It is semi-permeable. Okay, I've been told that the diagram that I was explaining has not been shown. So let me just, it has not been shown on the screen. So let me just show it again. So this is the diagram that I'm talking about, the diagram of the cell membrane. This is the diagram. If you look at this diagram, like I said, the protein, the cell membrane is made up of 3 layers. 2 of the 3 layers are protein layers, and one is a phospholipid layer. So the phospholipid layer is found, sandwiched, or it is surrounded between two protein layers. Now, as you can see, if you look at these yellow balls, the yellow balls at the top here, these yellow balls at the top here, they're the ones that shows the upper protein layer, and the one inside there shows the inner protein layer. And then in between here, we have got the phospholipid layer. So these are the three main layers. This whitish thing that you are seeing here is the protein layer. So as you can see, we've got various types of proteins here. But for you at your level, you only need to know that we have got proteins, the protein layer, both on the inner part and the outer part, and then in between here, we have got the phospholipid layer. And I explained what we mean by the word phospholipid layer. We say phospholipid layer because it is made up of a phosphate and lipid. So the word phosphate and lipid brought together form the phospholipid layer. Okay, great. So allow me to take you back to the other slide. Allow me to take you back to the other slide. Okay, Adan. Mohammed Adan. Yes.
In an exam.
Yes.
In an exam, can you be told to name the parts of the cell membrane?
Yes, you can be asked to name the parts of a cell membrane, especially if the structure is given to you. If the structure is given to you in an exam, then you can be told to identify the parts. So what you need to know is the basic structure of a cell membrane. The basic structure of a cell membrane is, make sure you know the positions of the two protein layers and the phospholipid layer. And then in between, we have got pores. Pores are tiny openings which are found on the surface of the cell membrane. They are very tiny. And because of the small sizes of these pores, they can only allow materials which are of very small sizes to pass through either into the cell or out of the cell. Okay? So we have got those pores which regulate the movement of materials into the cell and out of the cell. So it is important for you to note where we have got the protein layers and the phospholipid layer. Okay? So everything can be brought to you in an examination scenario. I hope your answered are done.
Yes.
Thank you. Great. So allow me to take you back to our slides so that you can continue looking at the functions of the cell membrane. Great. The cell membrane, now talking about the functions of the cell membrane, number one, it encloses the cell contents. It encloses the cell contents. And this function is contributed to by the three layers of the cell membrane, which gives it the strength to perform this function. Now we have said that it encloses the cell contents. The three layers gives it the strength to perform this function. The three layers gives it the strength to perform this function. Number two, the pores allow movement of materials into and out of the cell. The pores which are found on the surface of the cell membrane allows for movement of material or particles into and out of the cell. Now, in some cases, when in the next topic, which is the topic cell physiology, we'll be able to look at the cell membrane into details. And I just want to I want to mention something about it. When looking at the properties of cell membrane, you will find that it is said to be semi-permeable. We can say semi-permeable, partially permeable, or selectively permeable. Now, the reason why we say that is because of the pores that are found on the surface of the cell membrane. These pores are tiny. They cannot allow particles which are large in size to pass through. They will only allow those particles of the small size to pass through. Okay? So the pores allow movement of materials, of particles into and out of the cell. And remember, we also said that the cell membrane is made up of a phospholipid layer, which controls the movement of water, the movement of water-soluble and fat-soluble molecules, water-soluble and fat-soluble molecules into and out of the cell, okay? So the cell membrane, therefore, only allows small molecules like water and glucose to pass through, but not large molecules. So the membrane in this case is said to be semi-permeable. And on the word semi-permeable, you can add, it can also be referred to selectively permeable or partially permeable. You need to include those other two terms. So if you see the word selectively permeable, you can know that it means the same as semi-permeable. Or if you see the word partially permeable, you can know that it means the same as semi-permeable. Okay. So as we proceed, we now moving to the next cell organelle, which is the cytoplasm. The cytoplasm. Now, the cytoplasm is the fluid-filled medium. It is the fluid-filled medium in which chemical reactions take place. The fluid-filled medium in which chemical reactions take place. Now, inside the cell, we have got several chemical reactions that take place. We have got several chemical reactions that usually occur inside the cell. Now, all these chemical reactions take place in the fluid-filled medium, which is the cytoplasm, here. Okay, let me just find out what someone is trying to say here. Somebody raised his hand and he has left again. Now, inside this fluid-filled medium, which is the cytoplasm, this is where we have got all the chemical reactions taking place. Now, the cytoplasm, it is also where the other cell organelles are suspended. So that is why we are saying that it contains organelles and inclusions such as starch, glycogen, fat droplets, and other dissolved substances. Remember, it is a fluid-filled medium. In other words, it is made up of a fluid. And because of that, it contains, or it acts as a storage to these substances such as starch, glycogen, fat droplets, and other dissolved substances. And besides that, other cell organelles are suspended or are found inside the cytoplasm. So the cytoplasm is not usually static. When we say static, it means it is not permanent, it is not stagnant. But usually the cytoplasm is usually in a movement, or it is usually in a motion that is called cytoplasmic streaming. It is usually in a constant movement, which we call the cytoplasmic streaming. It is not static, but it has a movement known as cytoplasmic streaming. Now, the next organelle is the mitochondrion. Now, mitochondrion is a cell organelle. So the word mitochondrion here, if we call it mitochondrion if it is in singular. In other words, if it is one, we call it mitochondria. But if there are many, we call them mitochondria. Mitochondria if there are many. And remember, in biology, we always check even the spellings. We always check even the plurals and singular. If used in the wrong way, you can easily miss a point when you use plurals instead of singular, or singular instead of plurals. So kindly note that. So mitochondria if it is one, but if there are many, we call them mitochondria. So before we look at this, just want to remind you once again that in my slides today, I don't have the diagrams showing these various organelles. But when we meet for our next class, the first thing that I will show you is the different cell structures as seen under the light and the electron microscope, so that you can be able to identify where we have got all these parts, okay? So for now, we can just look at the functions, but when we meet, I'll be able to show you the diagrams of the cell as seen under the light and electron microscope. So talking about mitochondrion. So mitochondrion, or mitochondria if there are many, these are sausage-shaped cylindrical structures or grains which may be scattered about or concentrated at different points in the cytoplasm. Now, they're sausage-shaped. They look like sausages. They look like sausages if you look at them. So they can they may be scattered about within the cytoplasm, or they can be concentrated at different points. You can find them concentrated at one point within the cytoplasm, or they can sometimes be scattered, just found scattered within the cytoplasm. Now, the function is they are the sites for respiratory activities that yield energy to the cell. They provide site for respiration. That is what most of you usually say. They provide site for respiration. Now, when we talk about the word respiration is an active process that usually takes place within the mitochondria. And this is the process by which food substances are chemically broken down, either in the presence or in the absence of oxygen, to produce energy. You know, when you take in food, when we eat, the process of digestion takes place and the nutrients are released into the cells. So once in the cell, energy is produced from these nutrients. And the process that leads to the production of energy, is what is called respiration. So the function of mitochondria is to provide site for respiration. So respiratory activities that yield energy for the cell takes place within the mitochondria. So you need to note that the mitochondria is bound by two membranes. It is enclosed, or it has two membranes. We have what is called the inner membrane, and we also have what is called the outer membrane. So usually, the outer membrane is very smooth, while the inner membrane is usually greatly folded to form structures, other structures, which we call the cristae. And let me highlight the word cristae here kindly. Cristae. So cristae here, these are the structures which are formed when the inner membrane of a mitochondrion is greatly folded. So you can be asked, why do you think the inner membrane of the mitochondria are folded? The reason why they are greatly folded into cristae is to increase the surface area for respiration. Okay? That is the reason why the inner membrane of the cristae are highly or greatly folded. So another thing is that it has a fluid-filled cavity. This is still about talking about the mitochondria. It has a fluid-filled cavity where the respiratory reactions take place. I don't know whether I can be able to sketch a diagram of a mitochondria. Just trying to sketch it. I don't know that it will be very, very visible there. We have got the outer membrane there. So if you look at the inner membrane, they are folded. They look something like this. Very funny diagram on my screen there, but you'll bear with me. Now, it is something that looks like this. This is basically a simple diagram of the mitochondrion. So this part here, this outer part here, where I have my pen, this is called the outer membrane. Then the one that is folded, this one that is folded here to form teeth-like structures, this is called the inner membrane. So usually, like I said, the inner membrane, they are greatly folded. This teeth-like structure are called cristae. So the inner membrane here the inner membrane here is greatly folded into crystalline to increase the surface area for respiration. That is the reason why they're greatly folded. So inside here, in between the inner membrane, we have got a fluid-filled cavity, which is called the matrix. The matrix. So it is actually inside the matrix where respiration takes place. It is actually inside the matrix where respiration takes place. So the matrix here contains, Alex has got a question here and I'm trying to find Alex. Okay. Alex, are you there? Alex, I'm trying to unmute him. Okay, maybe you'd raise your hand again so that I can see you. In the meantime, you can continue. So this is inside the matrix. The matrix here, it contains some respiratory enzymes which catalyze or which increases the rate of respiration. Okay. Alex? Okay, let me try the other boy here. Yes, Stephen.
Is there a difference between enzyme reactions and respiratory reactions?
Enzyme reactions and respiratory reactions. Okay, great. Now, there's a okay. Enzyme reactions are those reactions that are catalyzed or which are facilitated by the use of enzymes. And respiratory reactions is an example of an enzyme controlled reaction. Okay, so there is a difference. Enzyme reactions are those reactions that are catalyzed using enzymes, and respiratory reaction or the process of respiration is catalyzed using enzymes which are found in the matrix of mitochondria. So I hope you get the difference. Okay, great. So the other person was Alex. Alex, could you please raise your hand if the question is still there so that I can let you ask the question?
Hello?
Yes.
Mwalim.
Yes.
This is a parent.
Yes.
Yes, I'm very concerned. This student, Keith Dungo, Alex Munana is also part of this group, but I'm concerned with Keith Dungo. I'm parent to Keith Dungo. He's not concentrating and it's a game kwa simu. Please punish that student. Wacha, mi sijui chenye nasema.
What is the name? What is the name?
Keith Dungo.
Keith Dungo?
Yes.
What is the class? Stream.
From 1 Southwest.
Keith Dungo.
Kazi yake ni kucheza game kwa simu na hasomi na ako on.
Okay, thank you. Noted.
Unmute me now.
Keith Dungo. Okay. I would urge everyone to take part in this and please try and concentrate. Try and concentrate. Okay, so let us continue. Now, having seen the structure here, and I want to make it clear to the person who raised the hand, the one who asked the question. If you talk about enzyme reactions, those are reactions that are catalyzed or are facilitated by the use of enzymes. I know some of you do not know what enzymes are, but more of the enzymes we learn when we get to the topic nutrition in plants and animals. That is the last topic in Form 1, a very interesting topic. Okay. Great. So allow me to continue before I finally give people the chance to ask the questions. Remember, our time is also up, and just let me finish up this. Okay. Now, the arrangement and number of the mitochondria in a cell depends on the cell energy requirements. the cell energy requirement. So in some cells, you'll find that there are those cells that require a lot of energy. And therefore, you'll find that because of the energy demand of these cells, there'll be a lot of there'll be a high number of mitochondria in these cells to provide the energy that is required. Now, cells which require large amount of energy contains very many mitochondria. Examples of these cells include, we have the muscle cell, the sperm cell, the apical meristems, which are actively dividing cells found in the stems of trees. And we even have the kidney cells, where a very important process of excretion takes place. So muscle cells, sperm cells, apical meristems, and the kidney cells. These are very important. These are cells where a lot of energy is required. And because of that, they have got very many mitochondria. They have got very many mitochondria. So I think we can, this is basically the structure, a simple diagram of mitochondria. A simple diagram of mitochondria, as you can see, we have got the outer membrane, the inner membrane, the cristae here, and the matrix. And I've been able to list their functions. So I would want to stop at that point without proceeding to the next organelle, which is the ribosome. But before I stop, I'd want to give time for the students who have questions. Alex Munene, I can see your hand raised again. Let me start with you before I move to Karaoke Kelvin. Okay. Okay, Alex Munene, can you hear me? Okay. Karaoke. Yeah. Karaoke Kelvin.
Yes.
Yes, you have a question.
Can we say that? Yes.
Yeah, I can hear you. What is the question?
Catalysts, you have said they speed up the process of respiration.
Yes, they do.
So catalysts in layman's are speeding. They are substances that help in speeding up an activity.
Yeah, we say they can either increase the rate of that reaction, or they can lower the rate of the reaction, depending on the specific functions of those enzymes. And the enzymes are the ones that perform the functions of catalysts. Now, I know that in chemistry, you learn about catalysts, and you also have catalysts in biology. But here, enzymes are examples of catalysts that help.
Catalysts.
Yeah.
Thank you.
Okay, welcome. The next question here is from Brevi. Brevi, are you there?
Yes, sir. I just want to ask whether you could please go back to the previous slide. I haven't finished writing the notes, kindly.
The previous slide before this, eh?
Yes.
Oh, okay. We'll just show that immediately as a. The next question here is from who?
Thank you.
Let me just find out who still have a question. Okay, Alex, I don't know whether you still have the question. If you have the question, could you please raise your hand again before I end the class? The next slide is here for Brevi, and plus another person, you're not taking that one. And just like I said I will try as much as possible and avail the diagram of the cell as seen under the light and the electron microscope, plus even some of the diagrams of this cell organelle, so that you can be able to see them and understand their functions. Okay, great. So I can't see any hand that is raised, so I want to believe that there are no more questions. And we want to keep on urging students to take this seriously, because it is very, very sad that a parent is complaining that the student is not being active, playing a game instead of concentrating in class. That is a very great sign of indiscipline, and we cannot tolerate that in a serious class like this. So take this seriously and make sure that you take part. Now, I want to give you some work that I will check in our next class, and I know better ways on how I will check this. I will easily know those who did the work and those who have done the work. So I want you to write down the following questions. I will just say them because I've not typed them here, but I will try and avail them to even the other platform so that you can be able to find them. Number one, state the adaptations of the mitochondria to its functions. State the adaptations of the mitochondria to its functions. State the adaptations of the mitochondria to its functions. Number two, outline how the cell membrane not how, but why. Outline why it is possible for the cell membrane. Outline why it is possible for the cell membrane to regulate the movement of materials into and out of the cell. Outline why it is possible for the cell membrane to regulate the movement of materials into and out of the cell. Number three, outline three structures which are found in plant cells, but not in animal cells. Outline three structures that are found in plant cells, but not in animal cells. Then finally, distinguish between the following terms. Number one, magnification and resolution. Great. So allow me stop there and see what the following have to say before we stop finally. Yes, this is who? Ben.
Yes, Chair.
Yes.
Chair, I did not get the first question.
The first question,
Yes.
The first question is, state the adaptation of the mitochondria to its function. State the adaptation of the mitochondria to its function.
Thank you.
Okay. Samuel? Samuel, are you there? Samuel, are you there? Samuel, could you please just send me the question so that I can be able to know it? Now I'm going to avail I will share with the teachers, your various teachers, the questions, and then they can post on your various WhatsApp platforms so that at least each and every person can get access to the questions. Okay. Otherwise, I want to stop there, and I want to wish you a good day. If there are any other questions, you can channel them on the WhatsApp platforms and you can be able to respond to them. So thank you and have a good day. Let us meet in our next class on Tuesday next week.